Thank you for the wonderful opportunity for me to present the data on the clinical activity of the RET inhibitor, Pralsetinib, also known as Blue 667, in patients with RET-fusion positive solid tumors. RET fusions are oncogenic drivers in multiple tumor types, and standard therapies have significant off-target toxicity in RET-fusion positive solid tumors, limiting their dosing and efficacy. Pralsetinib is a highly potent and selective RET inhibitor that targets RET alterations that include fusions and RET mutations, regardless of tissue of origin. So the data from this clinical trial, which is the phase 1 and 2 ARROW trial, have resulted in the US FDA approval of Pralsetinib for use in adult patients with metastatic RET-fusion positive non-small cell lung cancer. In addition, it is also FDA approved in RET mutant medullary thyroid cancer and RET-fusion positive all thyroid cancers. This study is still ongoing, and herein we present the data from patients with RET altered all solid tumors that enrolled in this ARROW trial. So what were the tumor types that were enrolled? The baseline characteristics of the tumor types enrolled were a total of 14 patients. And for GI ASCO specifically, the relevant tumor types are 3 patients with pancreatic cancer and 2 patients with cholangiocancer, in addition to tumor types like colon cancer, ovarian cancer, neuroendocrine, and thymic cancers. All these patients were heavily pretreated before enrolling on this clinical trial. The median age was 54, and 100% of patients received any prior systemic chemotherapy before enrolling on the study. The most common RET fusion partner was the CCD6, which was seen in 29% of patients. With regarding to activity and treatment duration of Pralsetinib in various RET-altered solid tumors, the objective response rate was 50%. The partial response was 50%, stable disease was 42%, and the best response of progressive disease was seen in 8% of patients. The disease control rate was 92%. Again, responses were observed in all patients with pancreatic adenocarcinoma, 3 out of 3 patients enrolled, and 2 out of 2 cholangiocarcinoma patients enrolled. Here, I'd like to share the story of a patient with cholangiocarcinoma who presented to my clinic. So this patient was progressed from all standard lines of therapy for cholangiocarcinoma, and her performance status was rapidly declining, and she was wheelchair bound when she was sent to our clinical trials clinic. And her tumor harbored the RET fusion. And because of this, we could enroll her on this ARROW basket study. And within a couple of weeks after starting on this treatment, she felt better. And her tumors responded to 64% shrinkage of target lesions of 20 months duration. And this patient went from being wheelchair bound to going on a ski trip to Europe in 3 months after starting this clinical trial. In addition, there was rapid clearance of the NCOA4 RET fusion transcripts in the ctDNA with rapid reduction in tumor markers. So what was the safety of this drug? The safety among the 14 patients with all RET fusion positive tumors, excluding the non-small cell lung cancer patients and excluding the thyroid patients, is consistent with the overall safety population. Again, the most frequent treatment-related AEs, more than 15%, were increased liver function test, namely the increased ALT and increased AST in 43% and 36% of the patients respectively. The treatment duration was 20.8 months with a median relative dose intensity of 98.8%. Most importantly, no patients discontinued the study due to treatment-related AE in this RET fusion positive cohort. So what is the take-home message? So Pralsetinib is FDA approved in non-small cell lung cancer and thyroid cancer that harbor RET alterations. Pralsetinib has broad and durable anti-tumor activity beyond non-small cell lung cancer and thyroid cancer, and has anti-tumor activity with multiple advanced solid tumor types. The objective response rate of 50% and disease control rate of 92% across various RET-fusion positive solid tumors is very encouraging, including response in difficult to treat cancers that progress standard lines of therapy like pancreatic cancers and cholangiocancers and neuroendocrine tumors. So after this data was submitted, we saw several rare tumor types enrolled on the study, like microcystic adnexal cancer, mesenchymal cancer, gastric cancer. Again, the important key message is to keep looking for these RET alterations in rare tumors and common tumors, because this opens up another line of treatment for these patients. Pralsetinib was very well tolerated. Its safety profile was consistent across the overall population. And in fact, this ARROW study is still ongoing and currently continues to enroll patients with various RET-fusion positive solid tumors.